These videos have been produced using content I have compiled into several ebooks, as you can see here. You can get these ebooks for free. If you are interested in getting your own copy emailed to you, click on the link below, fill out the form and it will be sent to you as soon as it is ready. Good luck in your exams.
All right, and welcome to our first review of past papers for the first time in, oh my God, yes, for this channel, right? And we are starting with a review of January 2017 for CSEC Bio as the thumbnail or whatever they call it, as it says. So let's get straight into it. We have our first question. We have here a group of students and they are conducting an experiment to investigate the rate of reaction. So this is looking at an enzyme activity experiment, and we're looking at the impact of temperature on the activity of this enzyme, right? So let's take a look. As those who are my students will know, the rule is before you even look at the question, if you are provided with a table, a diagram, a graph, you should take the time to make sure you understand the diagram before you go further with it. So let's take a look at what's presented to us. So as we can see, definitely we're looking at temperature. If this were to be a graph, this would be our x-axis because this is our manipulated variable, right? This is the variable that we are manipulating, right? And manipulated variables usually go on the x-axis if you were to draw a graph. And therefore this here, which is your responding variable, right? That would be on the y-axis, right? So on the x-axis, we have temperature. We have several temperatures here. We're going from 0 to 50 degrees Celsius. Now we remember that this here may be our optimum temperature, right? Optimum temperature probably. However, we are seeing that based on the rate of reaction, that it looks like the optimum was actually this one, all right? So this is probably the optimum temperature, right? And we see that from the rate of reactions, we go from 0 to a high of 24, then we drop back down to 0 really quickly, right? Note the units. So the units for temperature is degrees Celsius, and the units is milligrams of product per minute. So we are measuring rate in terms of appearance of product, right? Because when we are doing these kinds of experiments, there are two main ways we can collect our data. We're looking for appearance of a product, or we're looking for disappearance of a substrate, right? If the experiment is focusing on appearance of product, then you're going to get the typical enzyme temperature graph, right? However, if the experiment is looking at disappearance of the substrate, then the graphs are going to be flipped upside down, right? So instead of a nice peak, you're going to get an upside down U, right? So keep that in mind. If there are questions that have come up that use that type of experiment, right? So now that we've figured out what's going on with this table, let's take a look at the question. So the question is asking us to plot the data in table 1 on the grid below. So in the interest of time, I completed it already, but of course, let's just go through some of the basics as to what we want to, what we expect, right? So when you're doing a graph like this, all right, notice they didn't tell you what type of graph they wanted. However, anytime you're looking at the trends in something, so for example, we're looking at temperature and we're looking at how rate of reaction changes over different temperatures, then you want to get a line graph, right? So a line graph, you would use that when we're looking at trends, right? Change with respect to some other change over time, or some other variable, right? So we see here the some other variable in this case is temperature, right? We see that the rate of reaction is changing with respect to temperature. As temperature changes, rate of reaction changes. So in a situation like that, you want to do a line graph. And therefore we have our line graph here. One of the first things, well, we already figured out what the x-axis was. We had already figured out what the rate of reaction is, right? Note that we make sure to put our title for our axes, right? And what else can we see? We put our units, etc. Now, from what I remember with this paper, the way how the grid was set up, it didn't really give much space for a graph title. And especially given they just asked you to plot the data. So in a case like this, you'll probably get away with doing leaving out a title, right? But generally, as a rule of thumb, you should make the effort wherever possible to add a title to your graph. So for example, a title for this graph might be graph showing change in enzyme activity. With temperature, right? I need to do the temperature here, right? So that's if you were to add a title, okay? You would add something like that, okay? Especially for line graphs, that format of showing change in responding variable with manipulated variable, that works really well with line graphs, all right? So let's go on to the next part of the question. Now, before I come back to this, let me just outline how this will be graded. So it's 4 marks, right? So 4 marks, you're looking at formatting, you're looking at accurate plotting, all right? Okay, you're looking at things like 75% of grid. Okay, what else? Yeah, that's about it. It's 4 marks. So 4 marks will probably take up about 2 marks. Because format will be things like it's in pencil, you have your axes labeled correctly, right? With the units. Okay. The plot points, use that X or a dot and circle on X, right? These are the kinds of things that you would get marks for. So accurate plotting will be 1, 75% of it will be 1. Now, of course, what I'm outlining is a suggested mark scheme. based on a few decades experience teaching and working and having marked with CXC. But again, it's at the end of the day, it's up to CXC. But this also follows the kind of mark scheme that they give you for your SBAs and so on. So the kinds of mark schemes they use for your SBAs will be a template for what they would use in the exam itself, right? So that's basically how that will be marked. And then we get to the graph. They're asking you to identify the optimum temperature for enzyme X. So you're not going to go back in your mind and be like, well, according to the theory, according to what you would have learned off in a textbook, this is the optimum temperature. No, you're using the data in front of you. And as you can see, so I brought back the same graph, and we are going to highlight where the maximum rate is. So the graph gets to a peak at this point here. So this is our maximum rate. And then when you follow, you check to see what the value on the x-axis is, then we can see that this is our maximum rate. That's the temperature, right? So as we see here, maximum rate of reaction lines up with optimum temperature, right? So our optimum temperature is 30 degrees Celsius, all right? Then account for the shape of the graph. So they're asking for the shape of the graph between two sets of temperatures. So first they're asking you to look at between 5 and 30 degrees Celsius. So that's between here and here, right? So basically this area here, all right? This area, okay? So let's take a look at, if I were to do a quick, yeah, so this area here, all right? Good. So let's go to, what did we say for this one? It's with 2 marks. So I always, it's always a good, it's always best practice, I should say, to quote your data, right? Whenever you're talking about, you want to explain or describe, etc., a graph, parts of a graph, etc., even a table, any kind of data that's being presented, you should quote your values, right? So we have here, the first thing we take note of is that a description of what we're seeing, right? So we have a steady increase with temperature from 3 milligrams per minute at 5 degrees Celsius to 24 milligrams at 30. So we're quoting the values based on our, because we see here was about, it's about 5 there, right? So that's about 3, okay? And to account for it now, we would give an explanation. So first thing is that between 5 degrees to 30 degrees Celsius, you're starting at a very low temperature, and then you're going up to basically the optimum temperature for the enzyme. So definitely temperature would be your limiting factor here. This would be the factor that is dictating the rate of reaction, right? So that's another little point we can make here. So temperature is the limiting factor at this point, right? Okay, it is dictating right? It's dictating the pace of the reaction, right? It's determining how fast the reaction is going. So as the temperature increases, enzyme activity also increases. So why is that happening? Well, at a molecular level, what's happening is that as the temperature is increasing, the molecules are moving around faster, right? And therefore, there are more collisions between the enzyme molecules and the substrate molecules, right? And because of that, the chance of a substrate molecule colliding with an enzyme molecule and getting into the active site, if you remember how an enzyme works, right? So the substrate will get into the active site more often. And once the substrate gets into the enzyme's active site, then the enzyme can do its work, catalyze that specific reaction, the products would leave, and then will be free to connect with another substrate, right? So because of that, we have our increase in activity. All right, that's because of the more frequent collisions. Now, ideally, what we can do, I know I put 1 mark here, but this actually could just be 1 mark, and this could be 1 mark. So it can go in one of 2 directions. But I still strongly suggest that you open with some kind of a description of what's happening and quote some data. All right. Because again, it gives your answer a more what you call it gives it a better quality, and it gives the impression that you really know what you're talking about, right? And that's always a good thing to do when you are presenting these exams to your future grader, right? And yeah, and that's it for that part. So the next part now, they're asking about above 40 degrees Celsius. Again, we're gonna drop the data, right? We're just gonna say that it drops to 0 by 50 degrees Celsius, right? And then we explain what's going on. So above 40, enzyme molecules are vibrating so intensely, they can no longer maintain their structure, right? Because again, with high temperature, it's not just them moving around, zipping around in the solution. The molecule itself is also vibrating, because remember, the enzyme molecule is a very large molecule, right? So even though it's the molecule itself is a solid, basically, that doesn't mean it's not going to be able to move, right? So the molecule itself is going to be vibrating, and with more temperature, with higher and higher temperature, it's going to vibrate more and more intensely, right? And remember, the enzyme is like, it's not this solid like a piece of granite or something. It's basically this very long chain that's basically held together by different bonds, right? One example I like to use is, when you see these ladies with these big poofy hairstyles, right? That are really complicated, and they're held together with some hairpins, some strategically placed hairpins. And overall, it looks like this, solid structure. But once you take out a few hairpins, that's it. It's gone, right? It's disappeared, the hair flows down and all that, right? Same thing for, when you have different head ties, that certain types of head wraps, they are held together by some very strategically placed pins, bobbies, and of course, certain ways that it's tied. So the minute that you take out certain pins and you undo some ties, that's it, it falls apart, right? So same thing is going on with the enzyme. The enzyme is just really complicated structure held together, figuratively, by some strategically placed hairpins, right? Which would be your different types of bonds, right? Which you will learn about in CAPE. So imagine you take that and you start to vibrate that, right? Eventually, some of those strategically placed hairpins are going to fall out. And once they fall out, the entire thing falls apart. So that's what's happening to the enzyme molecule. It's vibrating at a high temperature. And as the temperature gets higher and it vibrates more intensely, those bonds that are holding the structure together start to break, right? And the entire structure of the enzyme starts getting compromised, especially the active site, because that's the part that we're the most concerned with, right? So the active site starts to change its shape. And remember, the active site needs to be a particular shape in order to receive the substrate and catalyze the reaction. So if the active site changes its shape, that's it, right? It can't work anymore, right? So at that point, that's when they say that the enzyme is denatured, and it is permanently denatured, right? In that you can't pull the enzyme molecule back together. It's not like if the temperature goes back down, it goes back to its original state. All right. Same thing. If the hairstyle falls apart, you know, just taking the hairpins and sticking it back in your head isn't going to bring the hairstyle back, right? So it's the same kind of thing, right? So it's denatured, it vibrates too intensely, broke down, and that's why the rate of reaction drops by 50 to basically 0. All right. And precautions. So precautions are basically, in an experiment, it is any action that you can take to ensure the accuracy of the experiment and reduce the possibilities of any kind of corruption or compromise in the experiment, especially when it comes to purity of the substrates, etc. Right? That's what the precaution is. It's just steps you would take to ensure accuracy and to reduce any kind of corruption of your substrates or products. Okay? And yeah, that's one of the examples, one of the precautions you can take. You can make sure that the substrate and enzyme mixtures are the same target temperature before you mix them, right? Because remember, this is a temperature dependent experiment. This is an experiment that's focusing on temperature. So if you take the substrate and the enzyme, and the substrate is at a different temperature than the enzyme, let's say your target temperature is 30 degrees Celsius, but your enzyme, you're taking it out of the fridge, so its temperature is 20, and the substrate, maybe you just mixed it up a little bit. Maybe you had to mix it with some heat, right, to get it to dissolve properly. And so therefore this temperature will be about 40. When you bring them together, the temperature will not be 30, right? It'll be some point between that of the enzyme and of the substrate. And even if you eventually bring it back to 30 degrees, remember that time you spent bringing it to 30 degrees, it would have been acting, yeah? So now you have a reaction in your boiling tube that would have been going on at a very different temperature than what you are checking for. So if you go and say, well, this is the reaction at 30 degrees, it would be inaccurate because the temperature really wasn't 30 for a good portion of the reaction, right? So that's why this is a very important precaution that shows up for these kinds of experiments. Any experiment that has to do with temperature, this is one of those precautions that's classic. You use that as a matter of course, okay? So let's take a look at the rest of question 1, right? So we have here questions asking, name 2 enzymes that break down protein. So any 2 of the following: trypsin, protease, pepsin, peptidase, right? You name it, you can name that and you'll be fine, right? 2 parts of the alimentary canal, aka digestive system, where enzymes break down protein, and that would be your stomach. That's where you find the pepsin and small intestine, where you find all the others, right? And let's see. Suggest the reasons why different enzymes for breaking down proteins are located in two different parts of the alimentary canal, right? So let's take a look at it from two directions. One is that the enzymes have different optimum pHs. If we roll back to these guys here, right? Pepsin loves acid, right? Pepsin works best in acidic conditions. Everybody else works well in alkaline conditions, right? And not even heavily alkaline, like, well, I should say, let me, yeah, they would be about, yeah, relatively alkaline. I wouldn't say more alkaline than in your mouth. Let's say that, okay? So we have here, because in your mouth would be like close to neutral, like 7.5. Whereas in the small intestine, you're looking at around 8.5, right? So because of that, the enzymes are going to have to work in different parts of the alimentary canal, right? You have your enzymes in the stomach, which is the structure of the stomach is designed to provide those acidic conditions as well as be able to tolerate those acidic conditions, right? It has a very specific structure to do that, you know, with all the mucus and the lining is constantly replenishing itself and so on, all right? So these acidic conditions, you cannot really host in any other part of the alimentary canal, right? So the stomach is there to make sure that the pepsin gets what it needs, right? And also without killing you, right? Without killing you, because remember it's very acidic. And that kind of acid, if it burns through into the rest of your body, you are gonna be in a problem, right? So that's why you have the stomach will have one type of enzyme in it. All the other enzymes can happily function in the small intestine. All right. The second reason is because protein molecules are really big, right? Very, very big. And therefore, it's not going to be as simple to break down as, let's say, some starch or some lipids, right? You're going to need a good few enzymes to break it down in stages, all right? And that's why we have several stages. So that's why with proteins, you'll notice we go, for example, when we look at starch is probably the biggest carbohydrate, right? Starch, the breakdown process goes from starch to maltose to glucose, right? Straightforward. However, proteins, you go from protein to polypeptides, right? So it breaks it down to polypeptide chains. Then you go to di or tripeptides. And then it breaks it down to amino acids, which is your final product. Okay, so you see how many more stages are involved. All right, so that is why you need several enzymes in different parts of the alimentary canal, right? So let's go to the next one, leguminous plants. And they are looking at leguminous plants. They manufacture proteins, nitrogen-fixing bacteria, etc. And they're asking us the type of relationship between the peanut plant and the bacteria, and that would be called mutualism. It's a symbiotic It's a type of, right, symbiotic relationship called mutualism, right? Then we were asked to explain the answer. So I always like opening, because explain is a higher level objective, right? And it usually includes lower level objectives. So if you see explain also includes describe, right? So you give a little description or definition of what it is you're going to talk about, right? You're saying that it's a symbiotic relationship. So therefore, you want to say what symbiotic is, what a symbiosis is, right? And then mutualism, what is mutualism? Because mutualism is a type of symbiotic relationship. So you give a little definition there, right? And then you describe what's happening with the peanut plant in that respect, right? We basically state that they have a mutually beneficial relationship, right? And therefore, that's why we can call it mutualism. And then we outline how are they mutually beneficial, right? The peanut plant gets the nitrates it needs, right? Without having to depend on what's in the soil, if it's present or not. And then the bacteria, they get easy access to nutrients, they get shelter, they also get ideal conditions. Because an interesting fact about these bacteria is that they're actually anaerobic, meaning they require an environment that is absent of oxygen, doesn't have any oxygen in it. So these nodules are designed in such a way to remove any oxygen from around the bacteria so that they can function ideally. So it's a really a very intense level of support that these legumes are giving these bacteria, right? And yeah, and that's your 3 marks, right? Let's go to, so we're looking at a food test basically, right? This is a food test. All right. And it asks to describe a suitable procedure for testing the presence of protein in peanuts, right? And so the first part is, of course, you have to prepare your sample, right? Always talk about sample preparation, because depending on the food test, we would prepare the sample in a different way. Of course, this is what you call your Buret test. All right, good. Then you're gonna add some, well, you have a liquid mixture now, right? So the liquid mixture, you're gonna add about 2 cm cubed. Yes, it is preferable that you give, you know, actual volumes, right? Actual numbers, right? So you give your 2 cm cubed of the sample to a test tube. Now, because you're not heating it, you don't have to use a boiling tube, all right? And then you're going to add 1 cm. So you see why it's important to use your actual numbers. You're adding 1 cm cubed of dilute sodium hydroxide. Dilute, please dilute. For the chemistry people, that'll be about 0.1 m, right? And you're gonna add about 1 to 2 drops of copper sulfate solution, right? So for the Buret test in particular, your numerical values, your volumes have to be precise, okay? And then you look for a color change, yeah? And those of us that remember, the color change will be from blue to violet, right? And then we have reasons why plants need protein. So any 2 of these, right? Growth of new cells, repair of cells and tissues, right? We want to make up components of cell parts, right? So if we're looking at cell membranes, we want to build up the cell membrane. We want to build up a vacuole, because remember, especially, well, not in plants, but in animal cells, vacuoles tend to wink in and out of existence. But in the case of the plants, yeah, you need your cell parts, cell wall, etc., right? You needed to build enzymes, you needed to make very important molecules like chlorophyll, right? Which is very important to a plant. So any two of those, and you're good to go, right? And you may come up with even more, right? So as you can see, all of that was question 1 with 25 marks, right? And this question tends to be the make or break question for many students, right? Because it taps into experimental knowledge and it's a lot longer than the rest of the paper, right? So that's it for question 1. So let's go to question 2. Right, so this one is really straightforward, right? You have to state differences between a plant cell and an animal cell, right? Well, I said any 3 of the following, but it's 3, right? So we have plant cells have a cell wall, animal cells don't, right? Now, notice that I don't just say plant cells have a cell wall, full stop, right? Because it is, when you see things like differences, when people say something different, this is what you call a comparison question. So you want to, so when you're making the comparison, you must always talk about the 2 things that are being compared. You don't just talk about one side, all right? So instead of just saying plant cells have a cell wall, full stop, you need to give the opposite of it for the animal cell, right? So plant cells have a cell wall is absent. Another way you can say that is you could add a little word saying like whereas, let me write it out. So plant cells have a cell wall, whereas animal cells do not. Right? And that's your answer. That's another way you could write your answer, right? So plant cells have chloroplasts, chloroplasts are absent. Plant cells have a large central vacuole. Animal cells have several smaller vacuoles. Note too as well that plant cells have a permanent vacuole, right? They have this large vacuole, and the animal cells, their vacuoles tend to be temporary, okay? All right, so that's another point of comparison, all right? Animal cells, in animal cells, the vacuoles show up as per needed, right? According to the need, okay? Then we have differences between a prokaryotic cell and a eukaryotic cell. Now, thankfully, this is as far as we go with prokaryotes and eukaryotic cells at CSEC level. Of course, we will go much deeper into it at CAPE level, right? You just need to know the differences between them, right? A prokaryotic cell would essentially be your bacteria, right? Essentially, right? Most of them are bacteria. Eukaryotic cells are basically your animal and your plant cells. All right. So that's a big way to tell the difference between them at this level, okay? So any two, right, of them, and of course 1 mark each. Big difference between them is that a eukaryotic cell has a nucleus. In fact, the name eukaryote comes from the, I think it's Latin or Greek for having some kind of a nucleus versus a true nucleus versus not having a true nucleus. Okay, so eukaryotic cell has a nucleus, whereas a prokaryotic cell does not. All right. In a eukaryotic cell, DNA is stored as chromosomes, right? So eukaryotic cell, the DNA is wrapped up in these proteins, right? And then organized into these chromosomes. So it's a lot more organized and compartmentalized, right? And in the prokaryotic cell, the DNA is just there, just some loose DNA just sitting in the cytoplasm, where there's this long loop, right? This large loop that's called a nucleoid structure, or plasmids, where these little tight little circles that you find in certain bacteria. Okay. And another big difference, as we said, they don't have compartmentalization of function. What does that mean? It means that they don't have their various functions in nice, neat compartments. All right, it's not as organized as a eukaryotic cell. So for example, in a eukaryotic cell, respiration would occur within a membrane-bound organelle called a mitochondrion, right? Whereas in a prokaryote, respiration would occur in these other structures called mesosomes, which is basically some membranes that just, folded in on themselves within the prokaryotic cell, right? So you don't have that same prokaryotic, you don't have that same organization that you find in eukaryotes, okay? There was something else, but I'll come back to it as I need to, right? So that's basically your prokaryotes and eukaryotes, okay? Oh, that's the point I was going to make. With another way that you can describe the difference is that prokaryotes do not have membrane-bound organelles. So let me just write that in. So prokaryotes do not have of course you would put the right now dot because you need to continue the point, right? Whereas eukaryotes do, right? Good. Function, again, for some reason, people tend to make this a lot more complicated than it needs to be, right? Cell wall maintains shape of the cell. Another thing you can say is that it prevents bursting under hypotonic conditions. Hypotonic meaning if the cell is in it's surrounded by a very dilute solution, right? Which means that water is going to flow into the cell and fill it up. So as we know in plant cells, because of that cell wall, the cell wall prevents the cell from bursting, from being so filled up with water that it expands until it bursts, right? That happens to the animal cell. That does not happen to the plant cell because of the presence of the cell wall, right? And then we have the cell membrane, and the main role of the cell membrane is just to control what moves into and out of the cell, right? It controls the movement of materials into and out of the cell. Then we have, next question, what happens when an animal cell is removed from the organism and placed in a concentrated sucrose solution? So we have here The focus, of course, is explain what happens. All right, so again, explain being a higher level objective, it is going to include in it a short description. You don't spend the whole question describing, okay? So you're going to state, that as part of your explanation, you're going to state what happens, right? So the cytoplasm, more dilute, water leaves the cell, down its concentration gradient, right? With loss of water, animal cell shrinks. Boom. All right. And that's the part where you say what happens. The animal cell shrinks. Okay. And that's basically what it is. Okay. And then we look at the opposite now. What happens when it's in distilled water? Okay, same thing. We explain that the cytoplasm less dilute than the surrounding distilled water. Water enters the cell, down its concentration gradient, cell volume increases, it expands and expands and expands until it bursts, right? Because the animal cell does not have that protective cell wall to hold everything in, right? And then we describe how it would be different. So again, this is another comparison question, right? Okay, so how would it be different if a plant cell was put through the same experience, right? Or the same conditions, right? So in the sucrose solution, so we're comparing, right? So you notice I'm saying, okay, well, as compared to the animal cell, water does also leave the plant cell, all right? And like the animal cell, it will shrink, but not as much, okay? But the cell wall cannot shrink, okay? And therefore the cell membrane is gonna tear away from the plant cell, eventually killing it, right? And that's called plasmolysis, okay? In distilled water now, water is going to enter again, just like the animal cell. Like the animal cell, it is going to expand. However, that cell wall is going to exert pressure on the cell. It's going to keep it from bursting. All right. And this is just another aside that eventually the pressure by the cell wall will actually stop the cell from taking in more water. Right? So it's not like it's going to be taken and taken and taken. Eventually, the cell wall is going to exert that kind of pressure on the cell so that it no longer takes in any water, right? And so that's question 2. A lot faster this time, right? And this is the final one for this video. The second video will be looking at questions 4, 5, and 6, right? So let's wrap up with question 3. And here we have components of blood, right? We have a nice little picture showing us different types of cells, and we are being asked to identify, right? So as per the rules, we are looking at this diagram. We want to make sure we understand what's in the diagram. So we know A most likely will be the platelets, because platelets literally are like fragments of cells. So it looks like fragments, right? So we're going to, say, okay, this most likely will be platelets, right? Just a quick thing. Now, the question is, between B and C, which one is a red blood cell and which one is a white blood cell? Well, we can say this one most likely is the white blood cell, because in fact, this would be like a lymphocyte, right? Because remember, lymphocytes have very large nuclei that take up the majority of the cell, all right? Whereas the red blood cell has this very, very specific shape on the middle. And that's because when you look at the side of it, look at it on the side, it has this sink, right? So if you're looking at it from this end, it will look like that in the microscope, because this part will reflect light differently from the outside, right? From the edges. So it has that particular shape. How do we know that it's not a nucleus? Because it actually doesn't have a nucleus. You'll notice that this particular spherical, well, oval shape. You'll notice it is located right in the middle of the cell, and it has a very strictly oval shape. All right. Whereas if you look at a nucleus has a little, it's not as neat, right? So for example, if we look at a cell, let's say this is a typical animal cell, right? A sink like that would look like this, a very neat oval in the middle, right? If it was this. However, if it's a nucleus, it tends to be more to the side, right? Unless it's like a lymphocyte, and it doesn't have that nice, neat oval shape. All right, it'll have a little, it'll have a little fringe, right? It wouldn't be a perfect oval, right? As if somebody drew it with a ruler or something, okay? So that's a big way, that's one way you can tell the difference and not get taken in by this and think it's a nucleus, right? Right, so we have A is platelets, B is white blood cell, C is red blood cell, right? And of course, the platelet is responsible for clotting, right? Then we have this nice little flow chart here, right? And whenever you get a flow chart, it's a good idea to almost read it like a story, right? Read it in sentences, right? So we have here it's saying clotting factors released from plasma proteins synthesized in the liver and circulating in inactive form. Prothrombin circulating in plasma is converted to thrombin. So this arrow could be what do you call it? It could be interpreted as is converted to. So is converted to, right? So prothrombin is converted to thrombin. Then we have fibrinogen with the input of thrombin is converted to fibrin, right? And that's how you would fill out your flow chart, right? Then we have a wonderful little genetics question, right? So we're asked to talk about our favorite condition, hemophilia, which of course is a sex-linked disease, all right? It's inherited and it's connected to gender. All right, we know sex-linked disease is not the same as an STI, all right? Okay, sexually transmitted disease, okay? So this one is inherited. And in this condition, the blood doesn't clot normally, all right? And the allele is on the X chromosome. So therefore, the Y chromosome does not have the allele, right? So we are being given a scenario where we take a woman with normal blood clotting ability and she's mated with a male with normal blood clotting ability as well, okay? So now notice in the diagram, they provide some of the information. Now notice that even though the female is normal, notice they give you a heterotrophic, hear me, heterotrophic. Sorry about that, heterozygous. A heterozygous genotype, right? And why? So that means even though she's normal, she has normal clotting ability, she is a carrier, meaning she is carrying the allele for the disease, but it's on one of the chromosomes. So she herself doesn't have the disease, okay? But she does carry the allele for it, okay? And they show us the gametes. Notice they write the gametes. You know, normally we'd write our gametes like this, right? So in this case, they're writing the gametes with the brackets. Whatever they use, you use it as well. So if their formatting is to put it in brackets, you also write it in brackets, okay? In fact, I kind of, you know, whatever, right? So you write it here. And again, in the diagram, they just wrote it out like that. So you write it out like that, okay? Now, usually with my students, I would give them a format to use if they are asked to do an entire genetics diagram, and you'd have a consistent format throughout. But again, if CXC is providing you with an incomplete diagram and you just have to fill it in, be guided by the format that they use, okay? And yeah, and there you go. You cross everybody, boom, 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 and you end up with basically this guy is going to be hemophiliac, right? A chance of the boy being hemophiliac. And the probability of having a hemophiliac child is 1 in 4, right? Because of all the children, notice, excluding gender, you have 4 possibilities, and of the 4, you have 1 in 4 is hemophiliac, all right? And what's the likely sex? It'll be male, right? And you indicate why, right? Sex chromosomes hold only 1 allele, and if it's present, there's no second allele to suppress it. Okay, so that's would be your explanation of why the child is what they call that, is male, right? And then we have why it's more susceptible. Well, think back to the importance of clotting, right? Clotting, the purpose of clotting is to close up any breaches or any openings in your skin, which is your first line of defense against any pathogens, etc., that is trying to get into your body, right? So if there's a breach of your skin, there's an opening, the blood clotting is a way to close back up that opening as soon as possible before infectious agents can get in. But if you're not clotting, your blood is not clotting, what happens? It gives these infectious agents enough time to get into your body, right? So that is why it's really important to be able to have your blood clot, right? And if you're hemophiliac, that means you are now even more susceptible to infectious diseases, okay? And that's what's being said here. And that's it for questions 1 to 3, right? So make sure to check out the second video that will go into questions 4 to 6. And because of course, those will be your structured essays. All right, so see you later. Bye.